Good morning. How is it possible that one human being can murder 20 innocent children while another individual can sacrifice her life in trying to save those same individuals? No one knows the answer, but it must be found within this 3 pounds of jelly-like material within our skulls. Enormous progress, as you know, has been made in understanding the organ systems of the body. We know a tremendous amount about the heart, the lungs, the kidney. But progress has been relatively modest in understanding the brain. Now, this is not because neuroscientists have not worked very hard at that issue, but it's because the human brain is so enormously complex, containing over 100 billion neurons. The liver has billions of liver cells. But what distinguishes the brain from the liver? In the liver, essentially, all the liver cells are doing the same thing. Whereas in the brain, most of the neurons have their own unique function. So therefore, the brain can actually be considered like many different mini organs. Now, unlike the liver, where most of the liver cells are essentially doing their own thing in isolation, in the brain, neurons are communicating with each other in very elaborate and complex ways. So, for example, one neuron can receive messages from up to 10,000 other neurons, and it in turn can send messages to 10,000 neurons. So we used to think of the brain as a computer, but it's more like a computer network with each individual neuron being a computer. So it's enormously complex. Today, we're going to begin to talk about the brain and how it works and how it fails. And I'm going to introduce you to the properties of neurons and neural circuits. A good place to start in talking about the nervous system is to talk about some of the major neurological and psychiatric disorders. This is not obviously a complete list of all the disorders that you need to know about, but I'm showing you this list to illustrate some, and in particular, some general themes that illustrate what you're going to need to know to understand neurological and psychiatric disorders. So let's just talk about some of them as well as their prevalence. So Alzheimer's disease, this is the big one. It's a loss of cognitive function and memory due to neuronal degeneration, in particular cholinergic neurons in the central nervous system. Currently, 4.5 million Americans are afflicted with this disease. It has a very low incidence for people less than 65. After 65, the incidence begins to grow very dramatically. Individuals 85 or older have a 50% chance of being afflicted with Alzheimer's disease. So if you come back here for your 60th reunion, assuming that some other disease does not get you, either the person to the right or the person to the left of you will have Alzheimer's disease. Epilepsy, brain seizures due to uncontrolled recruitment of electrical activity in nerve cells, 3 million Americans. Huntington's disease, a neurodegenerative disease. You're going to hear this word neurodegenerative coming up many times, associated with abnormal involuntary movements due to a repeated mutation in the Huntington gene. So here you have a genetic disorder of the nervous system. Multiple sclerosis, an autoimmune disease associated with sensory and motor loss due to demyelination. So I'm sort of emphasizing key words here. Myasthenia gravis, another autoimmune disease associated with muscular weakness due to loss of acetylcholine receptors at the neuromuscular junction, acetylcholine, the transmitter. Parkinson's disease, a movement disorder due to degeneration of dopamine-containing neurons in the substantia nigra basal ganglia pathway. Schizophrenia, delusions and hallucinations believed to be due to imbalances in dopamine and glutamate neurotransmitter systems. Stroke, a loss of specific functions due to occlusion of blood supply to specific brain regions. You add all these up, you have more than 17 million individuals with a neurological or psychiatric disorder. Not included in this list are anxiety and depression, which is about another 15 million. So you add all these numbers up, you have about 30 million neurological and psychiatric diseases currently in the United States. That's about 10% of the population. What are the general themes that you need to understand neurological and psychiatric disorders. You need to know about the genes that are related to specific neurological diseases that have a clear genetic basis, like the Huntington's genes. You need to know about the properties of neurons, like myelin, if you want to understand multiple sclerosis. You need to know about the properties of synapses, if you want to understand Alzheimer's disease and myasthenia gravis, for example. You need to know about the neurotransmitters. These are the chemical substances that allow one neuron to communicate with another. Given all these different neurotransmitter substances, like dopamine involved in Parkinson's disease, acetylcholine involved in myasthenia gravis and Alzheimer's disease, and their transmitter receptors. And then you need to know about the neural circuits, the ways in which different neurons are connected to each other to form these circuits, how these circuits normally function, and how they can fail in their function. So we want to talk about neurons and neural circuits. So let's begin talking about the neuron. Here is an idealized view of the nerve cell. The nerve cell is interesting because it's a polarized cell. Unlike a blood cell where you can't tell the top from the bottom, nerve cells are polarized, and they have 4 distinct regions. One is the cell body, or soma, and of course that's the region that contains the nucleus and most of the organelles necessary for basic cellular function. Neurons are also characterized by their long axon. It's over that long axon where a nerve action potential or impulse initiated in one portion of the neuron is transmitted to more distant regions of the neuron. And at these distal regions of the neuron are these specific specialized structures called synapses. It's at the synapse where the neurotransmitter substance, acetylcholine, glutamate, glycine, serotonin, you've all heard about these neurotransmitter substances. Then the 4th structure is the dendrite. It's on the dendrite where one neuron receives input or connections from another neuron. So let's look at one of these neurons once connected to a postsynaptic neuron. So we can define a neuron as being either presynaptic or postsynaptic. So this is the neuron that's presynaptic, and here's the postsynaptic neuron. So you see this neuron, the presynaptic neuron, making synaptic connections onto the dendrites of the postsynaptic neuron. And, of course, this one neuron, which is presynaptic to this neuron, is also postsynaptic to other neurons. So this neuron can make connections to this neuron, but it also receives connections from other neurons. In fact, it can receive connections from up to 10,000 different neurons. Here, based on this color code, you see just illustrated this one neuron receiving 3 connections from 3 different neurons. Okay, but think about the fact that this neuron can receive 10,000 connections from different neurons, and it in turn can make 10,000 connections to other neurons, only one of which is illustrated here. Now, what makes all this work, what allows neurons to communicate with other neurons, is this specialized structure called the synapse. And this next component of the slide shows a blow-up view of the synapse. A synapse contains a presynaptic component here and a postsynaptic component. The presynaptic component contains these vesicles, and these vesicles contain the neurotransmitter substance. Could be acetylcholine, could be serotonin, could be glutamate, could be dopamine. These vesicles then fuse when an action potential invades the terminal. It leads to the release of these vesicles through a process called exocytosis. The transmitter leaves the presynaptic cell, diffuses across this gap between the presynaptic and the postsynaptic cell, and it binds to transmitter receptors on the postsynaptic membrane, producing permeability and potential changes in the postsynaptic cell. Now let's talk more about the electrical properties of neurons, because that's the way neurons propagate information and transmit information. The first thing you need to know about is the resting potential. Nerve cells, like all other cells in the body, have resting potentials, and this can be illustrated by this cartoon in the next slide. Here's an idealized nerve cell, and outside the nerve cell is a so-called microelectrode. Now, a microelectrode is nothing more than a thin piece of glass that's stretched under heat to produce a very fine tip. In fact, the tip can be less than a micron or so. The electrode is filled with a conducting solution and then connected to a suitable recording device like this oscilloscope. When the electrode is outside the nerve cell, there is no potential recorded. But if you suddenly take this electrode and plunge it so that the tip of the electrode is now inside the cell, there's a deflection, a sharp deflection on the recording device. So now the recording device records a potential of about minus 60 millivolts inside negative with respect to the outside. And this potential is stable as long as the cell is not stimulated or damaged. This is the so-called resting potential. All cells in the body have resting potentials, including nerve cells. What distinguishes nerve cells from other cells in the body is that they are capable of changing their membrane potential for communicating and processing information. You remove the electrode, and then the potential goes back to zero. Put the electrode in, you get the resting potential. So here is an example of the consequences of stimulating a neuron. The idealized neuron is impaled with two electrodes, one to record the potential, and it's going to initially record the potential of minus 60 millivolts. This cell has also been impaled with another electrode, which for obvious reasons we're going to call the stimulating electrode. And this stimulating electrode can be connected to a battery, the polarity of which is illustrated here. And now we're going to talk about the consequences of closing this switch with different size batteries and think about how that is going to change the potential of the cell. Well, think about it for a minute. If we were to close this switch, we would connect the positive pole of the battery to the electrode, and that would tend to make the inside of the cell more positive than it normally was. And you might think that the amount that it makes it positive is going to be dependent upon the size of the battery. So we're going to show you a little animation where we sequentially close and open the switch, and each time the size of the battery is increased. That's the sound of the electricity. Okay, so we're changing the size of the battery, and each time we close the switch, we make the inside of the cell more positive. And when we make the inside of the cell more positive than it normally is, that's called a depolarization. Depolarized. It's less polarized. You can see that depending upon the size of the battery, the amount of the depolarization increases fairly uniformly until we get to a point called the threshold where we generate an entirely new type of potential, and that new type of potential is the nerve action potential or impulse. It's elicited in an all or nothing fashion. In stimuli below threshold fail to elicit an action potential. If the stimulus is at threshold or here, greater than threshold, you only get an action potential, and the size of the action potential doesn't change. Now, what does change, however, is the frequency of action potentials. Here, the stimulus was very brief, so there's only enough time to elicit a single action potential. What if we allow a greater amount of time? And that's illustrated in these four panels. Each one of these different panels has a different size battery. So here's the case where there's a very small battery. You can play the animation. The switch is closed. The battery is connected. The cell depolarizes, but the depolarization is subthreshold. Here's a somewhat larger battery. We're going to call this a weak stimulus. One action potential was fired. And that's it. A larger battery is going to produce a larger depolarization. Three action potentials, and still a larger battery. Sounds like a locomotive. Time here has been slowed down tremendously. If this was the real thing, you see this calibration bar is 5 milliseconds. These action potentials would have occurred very rapidly. The principle that's illustrated here is so-called the frequency coding in the nervous system. That is, the intensity of information is encoded not in terms of the amplitude of the nerve action potential or impulse, but rather in the frequency of those action potentials. Now let's talk about synapses. How does one neuron communicate with another neuron? So here's a cartoon showing a green neuron, which for reasons that will become clear in a moment, we're going to call an excitatory neuron, and it is making a synaptic connection to this postsynaptic blue neuron. If you produce an action potential in this green neuron, as shown here, that action potential leads to the release of transmitter substance from here, and that produces a depolarization, a potential change in the postsynaptic self. Transmitters released, permeability changes are produced here, and a potential change is produced here. This potential change is called an excitatory postsynaptic potential, or EPSP. Why is it an excitatory postsynaptic potential? Well, it's a potential recorded on the postsynaptic side of the synapse. Excitatory because it tends to depolarize the cell to threshold. There's the threshold. Now, in most cases in the central nervous system, a single action potential from one presynaptic neuron while it produces an EPSP in the postsynaptic neuron, that EPSP is insufficient to reach the threshold level. However, if two action potentials are fired in quick succession, then the EPSPs summate to reach threshold to fire an action potential in the postsynaptic neuron. Now, once you fire an action potential in the postsynaptic neuron, then that's the green light, and this postsynaptic neuron has an action potential that can then propagate along its axon and invade the synapses and set the next sequence going. So that's one flavor of postsynaptic potentials. There's two basic flavors. One is excitatory and the other is inhibitory. So this next slide shows you an inhibitory postsynaptic potential. So this red neuron is going to be called an inhibitory neuron for reasons that will become clear. It makes a synaptic connection with that same postsynaptic neuron. If you look at these synapses in a microscope, you'd be hard-pressed to tell the difference between this synapse and this synapse. But what they do physiologically is fundamentally different. So here was our excitatory neuron producing this EPSP. The two spikes here summated. Now, if you produce an action potential in this red neuron, and you look at the potential changes in the postsynaptic neuron, you see that that consequence of releasing the transmitter is not to depolarize the cell, but rather to make the cell more negative. That's called a hyperpolarization. So that moves the membrane potential away from the threshold. So this is called an inhibitory postsynaptic potential. Now, if you just look at this in isolation, you'd say, what's the point of inhibitory postsynaptic potential? Because the cell is not doing anything anyway, right? So why bother with inhibitory postsynaptic potentials or inhibitory neurons? Well, the key comes when you have both excitation and inhibition coming at the same time. The neuron has to make a decision what it's going to do. So here's where you can see really the effects of inhibition. Here is the IPSP produced by the one action potential in the inhibitory neuron. And these are the EPSPs that previously summated to reach threshold and fire an action potential. Now, if you fire an action potential in this inhibitory neuron before you fire the two action potentials in the excitatory neuron, now those two action potentials fail to reach threshold and fire an action potential. So this inhibitory neuron can control or affect the ability of excitatory connections from firing the cell. This process by which a neuron sums up the combination of excitatory and inhibitory synaptic connections is called integration. So we said that one neuron can make 10,000 connections to another neuron and receive 10,000 connections from other neurons. So there's an enormous possibility for different combinations of neural circuits. But it turns out that there's a relatively reduced number of circuits that seem to be recurring frequently in the nervous system, and we call these microcircuit motifs. And here's some of those microcircuit motifs. I'm going to talk about each one of these and give you a brief introduction to show you some of the interesting things that even these simple circuits can do. So here's one called feed-forward excitation. It's the simplest neural circuit. One excitatory neuron makes a connection to another excitatory neuron. We already saw that. Here is a circuit called feed-forward inhibition. An excitatory neuron connects to an inhibitory neuron, which in turn inhibits the next neuron in the chain. So this neuron is telling this neuron to go, but this circuit is telling this neuron to stop. Here's something called convergence divergence. These are just the words that conceptualize the fact that one neuron, this postsynaptic neuron, can receive multiple connections, convergent connections from other presynaptic neurons, and this neuron in turn can diverge and make many different connections to other postsynaptic neurons. Here's an interesting circuit called lateral inhibition. You see these excitatory neurons here. This excitatory neuron makes an excitatory connection to an inhibitory neuron on either side of it, and the inhibitory neuron in turn feeds back to inhibit adjacent excitatory neurons. Here's something called feed-forward or recurrent inhibition. An excitatory neuron activates another excitatory neuron, which activates an inhibitory neuron, which in turn comes back to inhibit the neuron that started the whole process. So this kind of circuit can be used as a braking mechanism, so to inhibit excitation in this chain. Here's an interesting circuit called a ring circuit. It's this recurrent inhibition, but one neuron is inhibiting this neuron, which in turn inhibits this neuron, which in turn inhibits this neuron, which in turn inhibits this neuron. Seems to make no sense whatsoever, does it? But I'll show you that this circuit can actually produce locomotive activity. Then there is, just as there is recurrent feedback and inhibition, there's also recurrent feedback excitation, and this can get complicated with ring circuits. So let's talk about what some of these circuits really do, not in isolation. And here's something that is quite important to all of you. And it's the basic feature of the neurologist exam when you test the stretch or knee-jerk reflex. This reflex is mediated by a simple feed-forward excitation pathway, as well as a feed-forward inhibition pathway. So when you tap the knee, you stretch the tendon and you excite sensory neurons that detect that stretch. Those sensory neurons then initiate action potentials in primary afferent neurons, the cell bodies of which are located in the dorsal root ganglion. Those action potentials then invade the spinal cord. They make two branches here. Let me just first talk about this branch. They invade the spinal cord. They release transmitter substance, excite the extensor neuron, and that in turn leads to action potentials which propagate out the ventral roots to cause the contraction of the muscle. So here is a simple feed-forward excitatory connection that mediates one of the simplest behaviors mediated by the central nervous system, the stretch reflex. This is also called the monosynaptic reflex because it's due to a single synapse in the central nervous system. There's also a feed-forward inhibitory connection here. So this neuron, the green one, excites the dark blue neuron. This is an inhibitory neuron, and what this neuron does is then release transmitter, which produces IPSPs in the flexor neuron, preventing the flexor neuron from firing. Why do you want to prevent the flexor neuron from firing? Well, if you want this reflex to work well, you want this muscle to contract and not this muscle. So by inhibiting this neuron, you decrease the probability of an inappropriate behavior. So here is a case where we're using those two very elementary circuits to produce this very fundamental behavior, the stretch reflex. Okay, we can also use the stretch reflex to illustrate divergence and convergence. So here's the basic system. A sensory neuron activates the extensor motor neuron that produces the chain, right? Now, let's look at divergence. There's not just one extensor motor neuron. There's a number of different extensor motor neurons, three of which are illustrated here. This one sensory neuron just doesn't connect with one motor neuron. One sensory neuron, the connections diverge, so it connects to multiple motor neurons. So there's your divergence. Where's the convergence? Well, there's not just one sensory neuron. There's multiple, hundreds of sensory neurons. And here's three sensory neurons that are illustrated, and each of these sensory neurons converge on this single motor neuron. So here's your convergence and divergence. Let's talk about lateral inhibition. This is really an interesting little circuit. And it turns out that this circuit is essential for a process called contrast enhancement or edge detection. Here are two rectangles. One that is dark and the other is light. I need to tell you that the illumination throughout the light one is identical and the illumination through the dark one is identical. But if you look at this carefully, particularly at this edge, you may notice that it seems to be a little bit lighter at the edge of the light region than the rest of the light region, and it seems to be a little bit darker at the edge of the dark region compared to the rest of the dark region. Does everybody see that? So the intensity here is identical throughout. The intensity here is identical throughout, but there seems to be a difference at the edge, and that helps us detect an edge. It's very dramatic here, but in lower levels of illumination, you might see how this could be important. Well, how does this work? This works through a process of lateral inhibition. Before I talk about lateral inhibition, let's talk about a very simplified version of this circuit without lateral inhibition. So here is the dark rectangle, here is the light rectangle, and we could code this in terms of the brightness of the light, say in lumens. So here the light is kind of, it's not very bright, right? And over here the light is much brighter. Now, this light falls upon the retina, and in the retina there is these receptors that convert the intensity of light into the discharge of action potentials. So let's just say that the dark region has 5 lumens of intensity, and the light region has 10 lumens of light intensity. So let's just say also that these receptors make synaptic connections, so-called second-order neurons. This is just a feed-forward excitation pathway. And let's just say the strength of the synaptic connection here is a +1, excitatory connection, +1, doesn't matter. As a result, 5 lumens of light intensity lead to an output of 5 action potentials per second. 5 action potentials per second here, 5 action potentials per second here, 5 here. Then you get to the light region. Now the light is more intense, and there's more action potentials because of the frequency coding process of the nervous system. So now the strength of the connection is the same. Now we have 10 action potentials here, 10 action potentials here, and 10 action potentials here. So without lateral inhibition, the perceived light would be actually the same as the actual light. You wouldn't see any edge detection or edge enhancement. Now, let's put lateral inhibition in the circuit. So each one of these receptors now not only makes a direct forward excitatory connection, but it also makes an inhibitory connection to the adjacent second-order neurons. So rather now, instead of producing 5 action potentials, it's producing 3 action potentials because it's receiving inhibition from the adjacent cells, right? That's a negative consequence of this process, but we'll see positive thing is much more important. Now look over here. Now the illumination of 10 produces 6 action potentials. It's less than what it was before because of the inhibition from this cell and the inhibition from this cell. But still you see that fewer action potentials here and a greater number of action potentials here, so you still perceive this as dark and this as light. Now the interesting thing here occurs at the edge, because now look at this neuron is only producing an output of 2 action potentials per second compared to the adjacent neuron 3. So this one would be perceived as darker, producing a darker signal, and this one is perceived as producing a brighter signal. This is producing a brighter signal because it's getting less inhibition from this neuron, and this one is perceived as dimmer because it's getting more inhibition from this neuron on the other side of the edge. So that's lateral inhibition. Now, lateral inhibition, interestingly, can also explain one class of phenomena called visual illusions. So sometimes what you see is not really what you're looking at. And there's a phenomenon called Mach bands. Everybody heard about Mach bands? Here are Mach bands. So what we see is a gradients of illumination. You see it's dark, bright, dark, bright, dark, bright throughout the field, right? Same thing here. Then there's this horizontal line. You look along this horizontal line, and what you see is a pattern of light, dark bands. These are the so-called Mach bands. Now, if you put a mask over this, however, just put a mask over this and just do this play. Now you see that the bands go away. So the illumination of this horizontal bar was the same throughout. You think that the illumination pattern is changing, but it's really not. It's an illusion. How can we explain this illusion? You can explain this illusion by lateral inhibition. Why is this dark? This is dark because this is a bright region, and the cells in this region are inhibiting the neurons in this region, so their output is less. Why does this appear brighter? This appears brighter because this is a dark region, and these neurons are less active, and thereby they're inhibiting the neurons that are responding to this illumination less. So lateral inhibition is good for edge detection, but it can also lead to some visual illusions. Let's talk about feedback or recurrent inhibition. And before I talk about it in terms of a neural network or a micro network, I want to talk about it in terms of a so-called nano network. So there's 3 levels of networks that we're going to be talking about. A micro network, that's these types involving a small number of cells. A nano network involving biochemical reactions in networks. And finally, macro networks that involve large connections or collections of micro networks. So a nano network of feedback inhibition is important for producing a phenomenon as shown here. Now, the standard textbook version of neurons is either they are silent or they fire action potentials when they are stimulated with a synaptic input. But some neurons, like this one, have a so-called endogenous bursting behavior. So in the absence of any input, this neuron is firing bursts of action potentials, becoming silent, firing bursts of action potentials again and becoming silent. So this is called an endogenous bursting activity. These kinds of activity patterns could be important for rhythmic behavior, like respiration. Well, how is it that we breathe in and out? This kind of neuron could be responsible for those kinds of rhythmic behaviors. How does this work? This works through a nano circuit. Here is an idealized version of that neuron, the outside of the cell, the inside of the cell. These cells have specialized channels that allow for calcium influx to occur. Calcium is a positively charged ion. It's in high concentration outside, low concentration inside. The feedback inhibition network, and I want to compare this network with this network. So the calcium comes in, and when calcium comes in, it depolarizes the cell. And as it depolarizes the cell, it brings the cell to threshold, causing the initiation of action potentials. But the interesting part here is that the increase in calcium also reduces the conductance of this channel to allow calcium inflow to occur, so the calcium inflow stops. But that calcium then, which is inhibiting the inflow of calcium, then is taken up by buffers, and so the calcium concentration is reduced, allowing the influx of calcium to occur again and repeating the cycle. So the point that I'm trying to make here is that this type of feedback inhibition circuit is capable of generating an oscillatory behavior in a neuron and an oscillatory behavior in a nerve circuit. This is a relatively short-lasting type of phenomena, short-term dynamics, once every 10 to 15 seconds. There is a subcellular network that's due to feedback inhibition that underlies 24-hour rhythms. And the big 24-hour rhythm that you all know about is the circadian rhythm. Circadian rhythms are due to oscillations produced in a special part of the brain called the suprachiasmatic nucleus. It's located just above the optic nerve. And what the suprachiasmatic nucleus does is it generates rhythmic patterns of output. And those rhythmic patterns of output have widespread effects on the release of hormones from the pineal gland, melatonin, regulation of the autonomic nervous system, the daily cycle of our body temperature, regulation of hormonal release. Now, it's interesting that this very complex and profound phenomenon can really be boiled down to a simple feedback inhibition circuit among one gene. So there's a gene called PER, and PER leads to the synthesis of the PER message and, in turn, the PER protein. PER protein then feeds back to inhibit the transcription of the gene. And this inhibitory cycle has a time course of about 24 hours. So the protein is made, it then inhibits the cycle, the protein is degraded, relieving the inhibition, allowing the cycle to happen again, making more protein, inhibiting the transcription, the protein is degraded, and the cycle repeats itself over and over again in a 24-hour period. Remember that crazy circuit with the 4 inhibitory neurons inhibiting each other? Well, that can explain, at least in principle, quadrupedal locomotion. So here's a little doggy showing the pattern of a walk. It's a gait, so-called gait, right? And a walk is associated with a movement of the left front limb, and a quarter of a cycle later, the right hind limb, and then half cycle later, the right front, and then the left hind. So the question is, how can this kind of gait be produced by the nervous system? Can a simple circuit do it? Another secondary question is that animals can do more than just walk. As shown over here, they can also trot. A trot is a gait associated with in-phase movements of the left front and the right hind, and with 180 degrees out of phase of the opposite limbs. But they can also do something called a bound, where the 2 front limbs are in phase and 180 degrees out of phase with the rears. And then there's something like a gallop, which is very similar to a bound. So we don't understand the neural circuits that produce these, but let's think about ways in which, in principle, you could do it. Here's all the dogs. You can get these animations online. So one way you could do it is as follows. Remember that endogenous bursting neuron? You could take 4 of those endogenous bursting neurons and have one neuron hooked up to the left front limb, another hooked up to the left hind limb, and so forth. And if you set up the timing just right, so you put these 2 guys in phase with each other, and these guys, hind limbs, 180 degrees out of phase with the first 2, you could, in principle, produce a gallop. These two limbs would go first and then would be followed half a cycle later by the hind limbs. Right? In theory it would work, but in practice there'd be problems because one is you'd have to set these up so they're exactly in phase, right, like this. And what would happen if there was some drift in the activity? So this one started firing a little bit earlier or later. Well, your problem would be that things would get very uncoordinated. So this doggy is not going to gallop. In fact, it's not even going to walk very well. So one way you could do this is with this circuit here. You take now 4 of these neurons, and each one becomes a bursting neuron. And when you do that, you get a circuit like this. And if you do a simple twist of the circuit, shown here, it's the same circuit. This blue one, that was that green one down here is now up here. Now this circuit, with just those 4 neurons, with those endogenous bursting properties, can produce each of those 4 different gallops with slight modifications in the strength of the synaptic connections between the neurons. So here's an example where this relatively simple circuit can, at least in principle, produce a fairly complex behavior, which gives us hope for understanding more complex behaviors in the nervous system. Here's a circuit I'm not going to go into. You can read about it. This is a circuit for the recurrent feedback excitation. This is important in the hippocampus. That's a region of the brain important in memory mechanisms where the memory is distributed among the strength of the synaptic connections. And then finally, we have macro circuits. All these different regions of the brain are highly interconnected to each other. And within each one of these regions are microcircuits, but this microcircuit is communicating with another microcircuit over here. And collectively, these circuits form something that looks like this. This is a macro circuit for the processing of visual information. You need to commit this to memory. No, I'm only joking. But just to illustrate the point of how complicated things are. So here, down here, processing of information from the retinal ganglion cells to the lateral geniculate up to various visual cortical areas. All these different cortical areas are communicating with each other through feed-forward and feedback connections. Okay, we have an opportunity to just ask a couple of questions. By the way, these are all in the electronic syllabus. Process by which a neuron summates synaptic excitation and inhibition is called. It's an easy one. Integration. Convergence is a process that contributes to it, but it's not the process. Do we have another one? The network motif. This would be a nano-network motif that underlies circadian rhythms. Temporal summation, spatial summation, feedback inhibition, feedback excitation, lateral inhibition. Absolutely. No lateral inhibition in that circadian rhythm circuit. Okay, let's stop there, and we're going to have a next lecture talking about how those synapses get formed.